Hi, everyone. This is Miss Moriarty, and I'm here to discuss with you our concept number 2 in our DNA and protein synthesis unit. So, here in starting with protein synthesis, we have to recall a couple of things back from the beginning of the year. So, we know DNA is a nucleic acid, and its main goal is to hold on to our genetic information. How do we get that genetic information? It is actually through the production of polypeptides or proteins. Now, thinking back to the beginning of the year when we talked about the building blocks of proteins, we said that its building block or monomer is called an amino acid. When multiple amino acids bind together, we call them a polypeptide. So, protein and polypeptide, they mean the same thing. Now, remember also, we mentioned that proteins are going to have very specific functions, and their function tends to relate to what their structure might be. Now, proteins actually have three levels of structure. They first form a chain of amino acids, that's their primary structure, followed by a secondary structure where they begin to bend, a tertiary structure where they now bend and continue to fold, and sometimes they can even bind with other polypeptides to form a quaternary structure. Now, putting this all together, we call this the central dogma of genetics. Now, starting with DNA, which we know is found in the nucleus, remember, it cannot leave the nucleus. But how does the code get to a ribosome in the cytoplasm? Well, it is a two-step process. First is transcription, which you can see in the image here. This occurs inside the nucleus. An mRNA copy gets made, and it travels out to the cytoplasm where we find a ribosome for proteins to be made. Now, that second process there, that's called translation. So in transcription, right, we make a copy of DNA, and then in translation, we turn it into protein language, hence why we call it translation. So putting that all together, DNA to protein, first starts with transcription, that turns it into RNA, RNA then gets translated into a protein. Now, there are three types of RNA that are involved in each of these processes. Now, the first type is called mRNA or a messenger RNA. This is that single stranded RNA that we've talked about. It's going to copy the instructions of DNA and carry it to the ribosome. Now, the funky looking molecule here is called a transfer RNA or tRNA. Here, this actually holds amino acids and brings it to the ribosome. And the last type is rRNA or ribosomal RNA. This RNA actually helps to build the ribosome and even form the peptide bonds between the amino acids. Now, in the first step, looking at transcription, remember the main goal is to go from DNA to mRNA, carry the code of instructions that DNA holds, and move it out of the nucleus. Reason why we need to do that? DNA cannot leave the nucleus. So where does this occur? That would be the nucleus. What do we start with? DNA. Once the process is complete, we end with mRNA. Now, what are the actual details of the process? That's what I'm going to talk about in the next slide. The first step, an enzyme comes in, very similar to DNA polymerase. We've got RNA polymerase. RNA polymerase binds to what's called DNA promoter region. So when we talked about replication, we said that primase lays down primers to prep the DNA to know where to go. Same thing happens here. So DNA promoter regions, RNA polymerase can pick up on where that is and begin to unzip the DNA. Now, how does it know where to go? It actually looks for a specific sequence in the DNA called the Tata box, or thymine, adenine, thymine, adenine. Once it picks up on where to begin, RNA polymerase starts to add RNA nucleotides. But remember, RNA doesn't have thymine. Instead, it has the nitrogen base uracil. So as it adds nucleotides, it follows the base pair rule. A now binds with U and C still binds with G. Once it is then completed, it will get released. DNA zips back up, and the mRNA leaves the nucleus. And so overall, what's the definition of transcription? It is DNA being copied and turned into an mRNA strand. Now, very similar, right, to replication. RNA is made in the 5 to 3 direction, and remember the DNA template's read still in that 3 to 5. So, for example, if we've got our double-stranded DNA, it's going to then split apart. RNA polymerase will then add in this direction here. But remember, it's only single-stranded this time. So once it's now made its copy, mRNA will then be ready to then be shipped out to the ribosome in the cytoplasm, while DNA then forms back together to create the double strand to remain inside of the nucleus. Now, in your note packet, what I'd like us to do next is practice actually transcribing a piece of DNA. So here is our DNA template strand. So this would, for example, be a gene that might maybe code for your hair color, for example. Here you're going to read the DNA in the 3 to 5 direction. So from left to right. And you want to follow the same base pair rule. A now goes with U and C still goes with G. So now take a moment in your notes and see if you can now transcribe this DNA sequence into an mRNA sequence. So at this point, it might be a good idea to pause the video. Now at this point, you should have paused the video. So now I will show you what your answer should have looked like. So this is what your mRNA sequence should have been. If we have a T in DNA, that means in the mRNA sequence, we will have an A, adenine. If we had an A, adenine in DNA, then that means in our RNA sequence, a uracil, U, will be added. If we have a C, cytosine in DNA, that means there will be a guanine in the mRNA sequence. And so as you went down the sequence, continuing to follow that same pattern, you have now made your mRNA and is ready to then be shipped to the ribosome inside the cytoplasm. All right, everyone, that's it for the video. Please make sure as you watch the video, you took notes, be ready to discuss this tomorrow in class, and please mark this assignment as done on Classroom.